Hi, Victoria. How are you?
I'm fine, thank you. I'm just driving home from work. How are you?
Hi, Koi. Welcome back. Thank you for coming.
Hi, Katarina, Victoria.
Great to see you
Thank you.
again.
Hi.
I will put up the link for the with the folder with the presentation and the movies in there. And so everyone, today
Yeah.
I'm putting up a folder link. So there are
Oh,
different
thank you.
files in there.
Thank you
And
so much. Now showing the drive. Yeah. Thank you so much.
yes, thank you for putting everything there.
Yeah.
We will start in around 9 minutes, so we have a few minutes in case.
Okay. Okay.
So everything seems to work. We can hear each other well. The link seems to work. So perfect. We're off
Yeah.
to a good start.
Yeah. Yeah.
How was your morning? Is everything good?
Yes. Now I in Bangkok, Thailand. I spent 7 months here. I I got used to the life here. Yes. And today, sunny day, and now the temperature is 27 degrees Celsius and will be around 30. It's
Oh,
tropical.
wonderful.
Wonderful. Yeah.
Enjoy it.
It's great. You have
Yeah.
endless summer right now.
Summer. Yeah. Interestingly, people here recognize winter and visiting shopping mall. We can find many advertisement showing winter sale, special discount for winter sale. But I have no idea what that exactly means, the winter. But the weather is always tropical.
So do people travel maybe then to winter places, or is it just is the mall really cold? Because I heard that in some quite rich places in Brazil and other places they put the
Uh,
mall temperature very cold so you can wear your winter clothes or your
maybe
sweater.
some people, yeah, some people may visit to cold, cold country like Japan. Many people travel to Japan. Yeah. Can you hear my voice clearly?
Yes, very good.
Yes.
Yeah.
Okay, because here sometimes internet connection became very bad, and now already showing internet connection is poor. The Clubhouse app showing me, but it can't help. So if you can't hear me well, please let me know.
Yeah,
Okay.
yeah, for now it's fine. I have that sometimes too, but it's still okay.
Okay.
So, for now it's good. We'll let you know, but thank you for letting us know. And please also meet Katie and Dr. Heidi. Hi, welcome. Thanks for coming. We will start in around 7 minutes. Thank you.
Hello, Katie. Nice to see you back. Hello, Dr. Heidi. I'm driving. I have the red bar too, so I don't know if people can hear me either. I know how you feel, Koi.
Yeah, we can hear you too, Victoria. It's it's working. So, thanks for being here. Yeah, Katie, she's in Australia, so for her it's is it already warm? Is it summer, Katie, for you?
Yes, it's getting warm. It's lovely.
One time I want to spend Christmas in Australia. It must be so nice. I heard it's like barbecue at the beach and it's a long break, so it sounds amazing.
Yeah, exactly. And my parents live right by the beach and the river, so we usually go and have like a picnic, barbecue down there. So not like the cold Christmas that a lot of the world is used to.
Yeah, that sounds lovely. So one day I'll be there. I always feel like reluctant because it's such a long flight. But yeah, one day I'll make it.
When the kids are less wiggly.
Yeah, I think they would be fine actually by now. Like they would be fine. And a good friend of mine, a really good friend of mine, he lives in Australia, in Melbourne. Um, and he's staying because he married and has kids now and everything. And I have to visit one day, so. So we will start in around 3 minutes. Welcome everyone, and feel free to share the room in the meantime to check out all the resources that are in the folder. There are several files in there that we will use during the talk today. So, and I'm sharing right now in the chat the link to the paper in case you haven't seen it. And yeah, this is a really interesting and amazing research here today presented by our guest speaker, Dr. Kuhei Mizuno. And yeah, feel free to share and we will start soon. Oh, Serena's also here. Yay. The whole crew is here, huh? Hi, Serena.
Hello. Hello, everyone.
You made it.
I did.
Yay,
Wonderful.
family
Good to
reunion.
And it's around the holidays.
Yeah, busy day, but I can't wait to hear the talk. Hey.
Has anyone pinged Dr. Shaw? I'm driving, so I'm not pinging.
Oh, let me do that. Thank you. I pinged few people. Oh, yeah, I did. I pinged her in. Yep. Might be late
Yeah,
for
it's
Dr.
late.
Shah.
I
I
know
don't
people
know.
have other lives. but, but sometimes I forget too. So
Yeah, she always tries to come whenever she can.
yeah, I like to be all together too. So I can't
Mm.
resist.
And she has always very interesting questions, so it's always great having her.
That would be a great end of year show. The most, well, not the most, excuse me, interesting questions that people have asked our guests that stick in our minds. Because she always comes up with really amazing questions, just slides in and just really has such an interesting angle on something that hasn't even been asked yet. So just think.
Yeah, I agree. Yeah, there are different highlights we could we could bring up. That would be really nice. I think it would be a nice thing to do. We should really organize that, to have like this end of the year summary come together thing. So yeah.
Just jumping to say good morning, good evening, good afternoon, any where in the world, because it's afternoon here. And want to thank you again, Katerina, for the beautiful insights, and suggesting that you have a highlights with of all the lovely rooms you've been actually doing lately with Victoria and Katie and Serena, because I love actually the content you offer here in Clubhouse. So I wonder, are you connecting in LinkedIn or any other platform, or just in Clubhouse? Because I always dream that you can form a great research of multidisciplinary sciencey approach with the nice way you communicate the science here in a very simple way. I think it's a big call for all of you to have a research together. What do you think?
Yeah, I mean that's amazing. Thank you, Heidi, first for the very kind words to all of us. And yeah, yeah, it's a great idea. And we've been actually creating some research. I know Serena came up with, ideas during discussions, and she started the research project, and I know a few other collaborations started here. So yeah, thank you for suggesting. And we can connect here, or yeah, on LinkedIn, or different platforms. You can just direct message me, and then we can discuss there then. And yeah, it's 9 o'clock, so time goes by. So we should start. So welcome everyone to Science Society, and of course a special welcome to you, Dr. Koei Mizuno. It's such an honor having you here, and that you take the time to present your really interesting research here with us. And before we start, let me give the audience a short introduction. Dr. Akoi Mitsuno got his degree and PhD in agriculture at Kyushu University, and his main focus or field is applied microbiology. And he started his career working on fungi and their amylases, and then he continued then transitioning to bacteriology using lactic acid bacteria, Staphylococcus, Bacillus, and many more. And his current interests and activity are in environmental bacteria, distribution, morphology, polymer production, interaction, and he also has a passion for research and discussion about the origin of life, artificial cell form life, and theoretical works in biology. So yeah, welcome. And before we start, usually Victoria asks a couple of interview questions, if that's okay with you. Okay, Victoria, the stage is yours.
Okay, thank you. I was waiting to hear yes, because I'm driving, so I couldn't see if I missed anything. But okay, thank you so much. And Kuwei, we are, as Katarina said, we're so glad to welcome you to Science Society and hear your presentation today. I have to say, when I was looking over your paper, that it's really a romantic beginning, because it starts. Oh, I'm sorry. Is somebody trying to speak, or is that background noise from me? We're good. Okay. I'm reading that the bacterium was found on a cave wall, underwater, under a submerged river on Honshu, and it's just such a beautiful image. And so, thank you for that, just for starters. And so, my question is, if you can consider back in your life and maybe childhood, where it was that you first developed an interest for science, if you can remember what was it that grabbed you and sparked that interest.
Thank you so much. First, thank you for having me, everyone. The society, Katerina, thank you so much, and Victoria, thank you for asking me a wonderful question. So, okay, so let me answer to the question from Victoria. Victoria asking the how I got interest in science and biology, in, for example, in childhood. Is this correct? My understanding.
Yes, that's great. And please answer however you choose.
Okay. Okay. In my childhood, in 1970s and 1980s, in my teens, we found rapid advances of biotechnologies. For example, when the first in vitro fertilization was reported, the media were eager to discuss the advances and challenges of biotechnology, which influenced me. Actually, my father was a medical doctor for women, so in my house we discussed such a science topic, and that influenced me so much. And in 1970s, 80s, we had a nice TV program for science. My favorite is Cosmos. The main anchor was Carl Sagan, the astronomer and professor at Cornell University. And my father recorded this program, and I watched the video many times. Such experience influenced me, and I liked reading. My favorite in my teens was Henri Bergson, a French philosopher. He published a book called Creative Evolution in 1907, which was later harshly criticized by supernatural and unscientific stance. Today it's called vitalism. However, as I understood some parts of his argument, he pointed out the limitations of a certain type of science at that time. A certain type of science treated natural phenomena linearly and assumed that everything was predictable. And he argued for the need for a new level of science. And later I came across a book written by the American theoretical biologist Stuart Kauffman. His theory, as I understood it, seemed to be an attempt to take a new stance on complex phenomena in biology. I became very interested in having the opportunity someday to reproduce such complex phenomena in my own experiments.
Thank you so much. It's so fascinating. We never know what path people took to get to the point they're at. And I'm so, well, first of all, I think many of us loved Cosmos and loved Carl Sagan and the enthusiasm that he brought to scientific discussion.
Do you know the program?
Yes, very well. Yeah, it's beloved to me and my family. I read his books and watched Cosmos and watched it again and again because it's not everywhere that you can. That watching that show is kind of like having him sitting across from you at the dinner table, and in a conversational way talking about science. with all his passion and all of the different directions that he would pull in to it.
Yeah.
So I'm really grateful to him too, as well. And I'm curious, how do you think that reading those two, the philosopher and the scientist that you mentioned, how do you think those influenced? I hear that it encouraged you to, it motivated you to work on complex ideas, and what you're about to present tonight is absolutely so exciting and I would say groundbreaking for us. But how do you feel that that philosopher influenced you in your methodology or your approach to your work?
Yeah. I think the experiments in biology field was quite different from what I thought before, like imagined before. So I did experiments. My career was uh, for focusing on uh, microbiology in industry. So quite different from what I aims in at my uh, young age. But today, if I had time, I'd like to show some perspectives uh, from philosophical aspects when I uh, did this study. I took care about.
Well, then let's get right into your talk. And just excuse me a moment. I would like to welcome Dr. Shah. Glad you're here with us tonight. And then pass you the mic back. And Kuwei, we are here if you have, for people who may be writing questions to you in the chat that we can share with you afterward and after your talk. And as well, I'm sorry, something happened on my screen. The houses. Yeah, we're here to facilitate a Q&A after you give your discussion, and so I will pass you the mic, and thank you so much for sharing what you've just shared with us.
Thank you. So, may I introduce the article?
Yes, thank you so much.
Okay.
And for everyone, the resources are in that Google Drive folder, so please click on it and access them accordingly. Thank you.
I'm a super beginner of Clubhouse app. I checked the internet, and this Clubhouse is a voice-based SNS, so people might listen to it without watching any materials. So I try to explain orally, but I'm not sure I can do well.
Wei, may I interrupt you for one moment? Even though it's audio, it's possible for people to click on what you've shared and then look at that and still listen along at the same time. And some listeners are listening on two devices as well, so they might, copy paste on a different browser. So please just share in whatever way feels most natural for you. Thank you.
Okay, thank you. So let's start. Thank you for introducing me. I'm a microbiologist. Today I talk about a new bacterial species I discovered in a Japanese limestone cave in 2008. So if you can access the, please access the article or material I shared. Certain bacteria are known to exhibit multicellularity. Multicellularity is a fundamental principle that led to larger size and higher functionality in life on Earth. But the process is not well understood. 2.3 billion years ago, photosynthetic cyanobacteria emerged in global outbreaks to bring oxygen to the atmosphere. In the oceans, sponges arose and are believed to be the origin of all animals, including human beings. But it's not known how certain unicellular organisms, single-celled organisms, combined and how they fixed their multicellularity as an activity in a population. Study might provide us an example of how a single-celled organism can evolve and acquire multicellular traits, and how a certain environment can involve the evolution. The summary is shown in the figure 1 of the article, and or in the material, the page 3 of the slide. Now, the cue for the discovery was beautiful iridescent hue of the colony of this species on agar, as shown in the figure 2 in the article. You can find a rainbow-colored colony. Agar contains nutrients for bacterial growth. Microbiologists spread samples on agar surface. Microorganisms grow and form a cell clump that we call colony. Normally, a bacterial colony on agar doesn't appear transparent, but opaque, turbid color because of the random accumulation of the cells without any ordering. I shared some pictures of iridescent rainbow-colored colony in the slides, page 5. And at that time, I felt there was something in these rainbow colors, but I didn't know what caused this. So I had suffered for years. I didn't know what does physiological means. But one day I tested a red fluorescent dye targeting lipid membrane on the iridescent colony and got an image showing some geometric pattern. The first photograph capturing the pattern of this colony is shown in the slide, page 6. You can find red geometric pattern on the black background. It's not shown in the article. You can see only in the shared slide. You see the triangular lattice-like pattern showed that cells in the colony elongated themselves to become filament in an ordered manner. In a moment, I felt I could almost understand why it's transparent, iridescent, and kept a thin layer, and why it can form a tiny, dark, turbid clump in a mature colony after some period of time. Some kind of control must underlie this structural change, and the controlled individual cell elongation should contribute to the ordering of the whole colony. Then proliferation starts again in some way in the colony. Then we started 2 experiments that focused on how a pattern in a colony was formed and how the pattern collapsed. The data of the article tried to show how the cells form a nematic liquid crystal-like pattern when the cell elongation is controlled as shown in figure 3 in the article, and how a cell clump proliferates inside a colony and is ejected from the colony, as shown in figure 4. We observed that after about 5 days, the short rod cells became crowded inside the colony, and after that came out of the colony, breaking the colony surface. You can find a series of pictures in figure 4 in the article showing the GFP green recombinant cells crowded out of the colony layer. The crowding out starts and finishes in a half day. The colony looks stable for about 5 days and nothing happens. And then suddenly this transformation happens. Usually microbiologists would take this as contamination. Another species are mixed in a same culture. I took this for a while, but observing it repeatedly several times, I noticed that the timing of the colony collapse was a phenomenon that could be reproduced accurately if we give a well-controlled culture condition. For example, we could say like this: today is Tuesday, so the collapse will occur around midnight of Saturday, like this. Then I thought that it should be associated with some ecological context, because the behavior looked so strange for a bacterium growing in a plastic petri dish with a controlled condition in an incubator in a laboratory. This species attenuates the growth for a while and then suddenly starts growing again, which looked generating a kind of pulse. The colony looked to behave in a pulsate manner. So then I tried to mimic a condition in the cave wall that located near an underground river. The wall was just above the surface of the river, about 50 or 60 centimeter high. We knew that this cave, including this wall, would be submerged by rising water from rainfall. So we try to submerge the colony in the water in a laboratory, in my laboratory. The rod-shaped cells were ejected from the colony. And to my surprise, those short rod cells spread very nicely into the water. The result, as shown in the picture and the video, in figure 4 and the video. That betrayed my predictions, because I had thought that the cells would be more adhesive and clumped together. Those data suggested that this species has a two-phase life cycle. In the first phase, the cells self-organize to build up the foundation of the colony architecture that appears to stick to the wall surface and to pack the daughter cells inside. In the second phase, the colony sets aside its growing and expanding for a while. daughter cells proliferate inside, and the colony ejects daughter cells when encountered on water floor, or the colony architecture collapses by proliferating cells. I drew the illustration of the life cycle in the article, figure 4, and the same illustration is shown in the material, page 9. In the illustration, I use the term germ, G-E-R-M, which means that the filament cells look like body cells, whereas the short rod cells proliferating inside looked germ cells. Two different functions with different architectures appears to constitute a multifunctional body. This is my so far hypothesis. I need more data to show the life cycle, but this is the outline of the article. Thank you.
Yeah, thank you so much for showing this beautiful presentation and your really interesting work with us. And yeah, I want to open up the room for questions. So please unmute. If you have questions, please share in the chat or raise your hand if you have questions. And yeah, and we'll go from there. So does anyone want to go right away or?
Yes, I can ask my question.
Okay. Go ahead.
So thank you so much. That was a very wonderful presentation, and really we need to go through by details. And my question from you, because we know the feature of the prokaryotes and we are aware of the self-regen expression, and also I was just wondering what type of cycle specifically did you find any specific cycle in your finding that was important to you? For
So,
example, did you
specific
find
did you,
ubiquitous amino acid or any kind of sources that was specifically through the transcription translation system that was interesting to you and you found it important?
thank you for Dr. Shah. What did you say? A specific what? Sorry, I couldn't follow.
Ubiquitous amino acid source that normally we are seeing in both prokaryote and eukaryotes. And in some of the prokaryotes, they have a different mechanism, especially when we are thinking about transcription translation system. And I was just wondering any specific cycle that you notice from your finding that it can be important as a source or for the, for example, engineering or enzyme engineering.
You mean you're asking the molecular mechanism that support this kind of life cycle behavior?
Yes, and you know that we have the
Yeah,
we can use the mimicking system and we can use the engineering for the enzymes. And I was just
yeah,
wondering about
yeah,
your
yeah.
opinion.
Yeah. So far, in molecular aspect, we read the whole genome, and now it's open in database, and we check several genes that might be associated with cell elongation, because we have a lot of reports about the molecular mechanism about cell elongation. Cell elongation is tightly associated with cell division system. Cell division system associated with chromosome replication. So we have checked the related genes about elongation, cell elongation. But so far we have no special finding, a new finding about the related genes about cell division and elongation. Uh, we have several knowledge about a specific site cause elongation, but as far as we know, in the molecular level, we have we have not yet identified some related genes, enzymes that could explain this phenomenon, elongation and life cycle. So
I
this
see.
is, yeah, yeah,
Yeah,
yes,
I
but
think I got my answer. So, yeah, as you just mentioned, that sometimes it's very attractive for engineering because they are using the self-region expression, and you just explained about that. So, I'm passing the mic to the next person.
Thank you. Yeah, go ahead. Sorry, nothing.
So yeah, very fascinating talk. In the movies, I went through the movies, and
thank you so much.
um, just a beautiful presentation. And when you're looking at, so the streaks developed this, rainbow character. Um, but in terms of the colonies going through the multiple stages, first the crowd out, and then the ejection into the water. And you noted that there's a specific timing to that. Were you able to identify? And I'm thinking about, in this underground cave, and it's sometimes submerged and sometimes it isn't. Were there? Do you noted? Did you find specific triggering events from the environment that would induce these changes? Or is the timing somewhat related
Yeah.
to that? Mhm.
You're asking the uh, why we study in a cave and how we noticed such a
Well, more so the organism adapting to that environment, and is the timing of its life cycle stages related to, any environmental factors that would drive that?
uh, okay.
I mean, I don't know if you found that, but it's kind of a curious question.
How we could distinguish this species behavior from others because we have several examples that shows similar multiple uh, behaviors like fungus or in prokaryote bacteria we have some examples that shows multiple life cycle. So how we find and distinguish from others? You ask me.
I'm just curious about
Yeah.
the specific adaptations of the species to that environment, and whether you found any particular coupling that would drive its life cycle there.
Yeah. Honestly, it took long time. For example,
Mm.
I I I isolated this species and first I identified genetically. It's very quick and simple today, and this is new species. I knew in 2008. This is taxonomically, genetically new species. Oh, I can publish one in the taxonomical journal. So I noticed this species in new in 2008. But after that, I found the elongation and the pattern formation in I found this in 2017. So 9 years already passed, uh, and so it's very hard for me to notice this is something different, because the normally microbiologists observe cell shape by microscope using liquid culture cell. It's a simple. When I when correct microorganisms and cultured in a test tube and then observed by microscope. I observed this species by uh, optical normal microscope and electron microscope, and all data showed me this species. The shape is rod shape, normal short rod shape. The first image got into my mind so firmly. So this is short rod cells, how they generate this pattern and iridescent colony. So I hypothesized in the beginning I, that the cell excretes some kind of materials. That material might cause iridescent colony. So I tried to extract and purify unknown substances excreted from the species for 5 years more, but
Mm.
I couldn't extract anything. And one day, almost accidentally, I observed directly colony on agar. It, I we don't usually observe like this. That that's a cue of discovery, and it took 9 years. Yes,
It's amazing. Such persistence, but it really produced a fascinating finding.
thank you so much. Yeah,
Yeah,
and
thank you so much. So do you think it's necessary, because, in the paper was mentioned that the environmental conditions are quite important, I think, for this behavior or organization to happen. So you think that's the most important factor, that it was found in a stream of water, or do you think this is also possible in other environments, it's just we didn't discover them yet?
mm. Yeah, 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 yeah. I think this is related with perspective, how we see phenomena, with what kind of perspective. Some people pointed out in internet. This article is interesting because focusing on the ecological context. Uh, Honestly, I didn't know well before I published this, but during the review of this article, manuscript to eLife, editor and reviewer pointed out this aspect, and I checked the situation in evolutionary biology, and people discussed about what important for for a certain evolution. Evolution involves endogenous and exogenous selection process. So then the one mystery is what driving forces acted to achieve this evolution. Example, multicellularity, when single-celled organism became multicellular. There is such a discussion today. I didn't know. There are two problems. First, if some useful genetic mutation has occurred in multicellularity, how is this mutation fixed in the population of a single species? And I came across a theory by Professor Black of the University Adelaide and Professor Rainey of the Max Planck Institute. They proposed a theory called ecological scaffolding hypothesis for this problem. They published a pretty big manuscript article in 2020. In the same year, I posted my preprint to the bioarchives. I noticed the theory, but I didn't understand well, deeply. The ecological scaffolding hypothesis, as I understood, points out the difficult part of this problem. Scaffolding is a temporary support structure that should facilitate a certain evolution. For example, just like after an animal crosses the surface of a frozen lake, once spring arrives, the ice melts, there will be no evidence to prove the crossing on ice. As such, we tend to ignore such temporal support structure that might have once existed in a certain environmental context. The hypothesis is that we can explain the drive toward multicellularity by assuming such support by the environment. That's the point. I described some in my article, but I gradually noticed this kind of discussion ongoing today. And the second problem is how can the process towards multicellularity be stabilized in the intermediate state before multicellularity is achieved without a reversion to a single cellular life in the beginning, very beginning stage of the evolution, in the terms of the need for a mechanism to prevent reversion. This is another discussion, and I noticed Professor William Ratcliffe of Georgia Institute of Technology pointed out this problem by proposing the concept of ratcheting to address this issue. These theories, I checked before and after submitting my article. I noticed and understood in my own way and explained as much as I could. This is a background.
Yeah, thank you. That's really interesting, and it's a really interesting discussion. And do you think that a similar mechanism was responsible for multicellular organism origin in not in bacteria but in organisms with a nucleus or cells have a nucleus?
You mean you asking there is another similar bacteria, right?
No, no, I'm asking also if in non-bacteria, do you think that the origin, the first multicellular organisms, do you think they used a similar mechanism to when multicellular organisms started? Do you think it was also in this under the same environmental factors and in a similar manner first, and then it continued evolving?
Of course, I don't know, but I believe ecological context is should be taken more. How should I say? It's important for the beginning of the emergence of multicellularity on the Earth. Environmental support might have take more important role than we think now. I believe so. And in a cave, I don't know. This is one example in a specific environmental condition. So how we have no idea how the first multicellularity emerged uh, in what kind of environment. Uh, we don't know, but we I believe we can we can think more freely if we take environmental support, consider environmental support into the emergence of a certain kind of evolution. In the case of not only in the case of multicellularity, but also in the case of other evolution. So yeah, I wanna talk about this. Some people argue that in internet community like this, this paper about this paper, whether or not the strange nature of this species can be regarded as multicellularity. People doubt this species should be regarded as multicellularity because, as they say, according to the current definition of multicellularity, different cell types must be dependent on each other. So someone pointed out that by the current definition, H3 is not, this species is not multicellular. But I think we can think more freely about the evolution of life if we can consider environmental support. And this study is not trying to define a new multicellular bacterium, but it's a study on the process of how living organisms evolved from a single cellular to multicellular. So I want to talk about whether definition is a good approach on the origin problems in biology field, such as origin of multicellularity or origin of life. When we try to define something, that means that we try to distinguish which properties of a species meet the definition and which properties don't. Whether or not such distinctions work well depends on how much we know about the things we have definitions about. For those things that we have sufficient knowledge about, we can expect that definition works well. For example, when we try to define a new species as a member of existing multicellular organisms that we have today, which is easy because we know well enough about existing multicellular organisms. The definition-based approach works well. What about the case of things that our knowledge is fatally lacking? For those that our knowledge is lacking, a definition-based approach would not work well. We should consider if it works well for the origin of multicellularity or other origin problems in biology field. First, I think we should recognize that our knowledge of origins of multicellularity is fatally lacking. The emergence of multicellularity may be billions of years old, and we don't know how old it is in the first place. We should be aware that it's very unreasonable to ascribe current properties to such a past process. Describing a multicellular organism by using the list of the property of the present organisms, which is termed taxonomy. But this work is not taxonomy work, but origin of multicellularity.
Curious on a follow-up on that point. You mentioned that you weren't able to identify the signaling molecules that were expressed that that could drive the organization of the spatial domains, and then it would be an interesting um, question if those signaling molecules were identified, if the spatial domains, if the cells within the different spatial domains were, then consequently secreting corresponding signals that, what they weren't all uniform, but they differentiated across those domains for separate functions. That would seem to indicate that the colony is operating in at least a coordinated multicellular sense. And I could see your point about how it's exemplary for multicellular behavior. Do you have any thoughts on that, or?
You are talking about timing of transformation of the colony.
Well, does the in the direction of a multicellular organism,
Yeah,
would there, in one stage that's driving the organization, the liquid crystal-like organization in these different spatial domains, do those
yeah,
spatial domains then take on different expression profiles that serve the function of the overall life cycle of the colony? That would be in the direction, I
yeah.
would think, toward the multicellular behavior.
Um, I have no data about honestly chemicals, but there should be some signals, chemical signals excreted from one colony and influence adjacent colony, and transformation happen like a chain reaction we observed. And some chemicals should regulate the cell formation, elongation, and and let the cell enter division phase. So several different kind of chemical signals should involve in this multicellular life form. Yes, this is another study we need.
I suppose it'd be an interesting experiment if during as the spatial domains form, if samples were taken and reseeded, were those cells in different states, or did we, or do they reset and reform the entire colony, or is there some period of remodeling
Yeah,
where that would occur?
yeah, yeah. I agree.
Yeah, thank you for the question. I see that Jake joined us. Jake, did you want to ask a question?
Sure. What kind of food does this cell take? Is it anaerobic, uh, or what does it eat?
Sorry, the internet is here. Internet poor. I can hear you well. Please, once again, please. Sorry.
What kind of food does it eat?
Food.
Yeah. What does it eat? Is it anaerobic, or what does it do? What does it do?
The you
What is it? Does it have a mitochondria?
are you you're asking the culture condition for this species?
Yeah.
Ah, culture condition is aerobic, um, and we we did a series of taxonomical experiments for this species, and we identified that this is absolute aerobe. So we can't culture this species in a anaerobic condition. We observed. Yeah, is this okay?
Yeah. What in the mine that it was in? What was the environment like? Was it highly mineralized? What was it like? Was it acidic?
In a cave, in that cave.
Yeah, yeah.
Yeah, yeah. This is a limestone cave, so and I isolated from dripping water on a cave wall, and the water, it's a calcite rock, so the water, the pH of water is alkaline about around 8.5, and this species prefer alkali site to grow. They can't culture at acidic site pH, and the temperature is pretty low. The about inside the cave uh, around 15 degrees Celsius, and the optimal temperature for this species is also relatively lower. The optimal is around 20 Celsius. That is relatively lower compared to the cross relatives of this species. So,
So it would be fair to say that this could be conditions that were similar to the beginning of life, the 3 billion years ago. Then, correct?
No, this is just specific condition for this species.
Okay.
May I chime in here? This is Michael. Okay, so the definition of life. High school students, read this. They're able to take in food as nutrients, and they're able to have waste products. But more importantly, with all the clubhouse I've been involved in, artificial intelligence. So life being able to reproduce, have a free will. So my definition of life is not just taking in food, nutrients, and waste products. Now, the other interesting thing is animal, mineral, vegetable. Mineral, I'll take it out. They're non-organic. Animal, mineral, yes. Do plants have a decision-making capability? Well, I've been studying for the last 6 months quantum biology. So I don't want to take the time of the group here, but artificial life is the topic. Artificial intelligence is different because software could be fooled thinking that that thing could play chess. Okay. I don't want to take this group into the clubhouse groups of artificial intelligence, but I do not think artificial life is on the horizon because forget my philosophy of uh, creation and evolution, but I don't think we are here to create truly artificial life. Now, biologically, we can create artificial livers, artificial hearts, we can do stem celling. Biologically, on the engineering side, believe me, I've been involved in many things. That's not artificial life. That's just like you can say your fingernail cuts off and you grow back. Cut off your arm, well, it might not grow back. So I'll leave that to all the doctors in the room. But thank you for listening to me.
So I was just trying to find out why you connect it to the beginning stages of life before it was when a cell was just like one cell and didn't have multifacets to it. I'm sure you've already said it, but I think I might have missed it.
Well, the beginning,
No, I'm sorry,
one could go back.
not you. I was talking to the person who's giving the
Our
speech.
guest, yeah,
Oh, sorry.
thank you, Jake. Yeah, we're trying to bring friends up from the audience to address our guest with questions. We're so fortunate to have him here, and so this room is for listening to our guest's presentation and also to ask questions of our guest. It's not each of our personal rooms. So thank you so much, Jake. And Kuwei, that was a question for you.
Thank you so much. I'm enjoyed listening to wonderful talk. I can't listen in a academic conference. I really enjoy. So what should I answer to?
I was wondering why you connect it. What is similar to this cell that makes you think it has something to do with original cell biology, like what before cells had multifacets to them?
You mean you're asking why?
What does this cell do that makes you think it is like the cells that were 3 billion years ago? I'm just trying to connect why there was the connection to cells. Uh,
So the discovery here is that bacteria organize themselves as kind of a multicellular organism, and this was not recorded ever before. And we are talking about how that comes, like which environmental factors could produce this
okay,
And
behavior. So
I
please,
see.
yeah.
this is happening in that cave now. the whole process of multicellular organism. But I was surprised at the situation of the research about the origin of multicellularity. And so this example might fill the gap and lacking of our knowledge about the origin of life, particularly the process, how ancestral single cell can behave and come together and form a clump and evolve to multicellularity we can recognize today. There is some we can find several multicellular organisms, but it's already completed, already perfectly organized organism. But we have almost no data about how they evolved. So this is one example to fill the gap about how an ancestral single cell organism can evolve to the multicellular form.
Well, with
So,
all due respect, if they took in nutrients and they took on a process and they had waste products and they multiplied, that's the simple biology that every high school student learns about being
I'm
an
sorry, but
organism.
you're not
Okay.
understanding the point of the talk, and I invite you to read the
Yeah.
article or
So,
yeah,
so,
maybe read the
so,
article first.
so, so my question again, sorry to take over, but I'm trying to grasp it. So these cells act as though they're multicellular, like these cells can come together as though they're one body. Is that what you're saying?
Yeah. And
Oh, wow.
on the way to multicellular form.
Wow.
yeah, what's interesting, an interesting, point about this is that, and in what would be critical to show that it is acting as a sort of proto-organism, is that as the colony grows, that the cells would differentiate and serve a function for the whole that would be distinct, as opposed to continuing to be individual cells that act randomly and according to just their own life cycle. That you would get differentiation across spatial domains that would have distinct function to complete the life cycle. That would at least indicate that it's along the lines of acting more in an organism capacity, I would think. And it's fascinating to see such an example documented.
Yes.
Yeah. So when it does make one specific body, like when you said that it sort of pushes, it protrudes from the mat. Um, what is the thing, like the wall that is formed, like how cells have cell walls, and that separates them from other cells, and then organisms have skin, and that separates them from other things. um, and is it possible for this thing to just like appear and then disappear, and then the cells that were inside of it are still alive and can do that again?
We are talking about prokaryote, Jake. Please read the article first.
Okay, my bad.
So it's about bacteria, right?
Yeah, bacteria.
It's even in the title, a new type of multicellular
Bacteria.
bacteria.
Yeah.
Yeah. So
Yeah.
yeah, the title of the room.
This is a
has it.
bacterium. Yes.
But that
Um,
the when it goes into the second phase of its life cycle and starts ejecting, there's a a regionally specific uh, place where that it ejects the new daughter cells into the environment. It's not it's not every cell for themselves that just continues to replicate into a mass, but there's an organization to it. And
Ah, that's the answer to my question. That's exactly what I was questioning.
Mm.
so that's interesting how that organization is encoded, and it manifests through a collective behavior
Mm.
that is not an individual cell's behavior. It's a collective, and
Correct.
it's spatially and regionally specific.
Yes. Corrective behavior.
and also, the form of the behavior is also really interesting, that you described in the paper, um, that it's like a liquid crystal, like the properties of this layered structure colony behaves like a nematic liquid
Yeah.
crystal. That's really fascinating to me. And is there, like, could you maybe explain for the audience, do you think it has properties to be organized that way that
Yeah. I'm a
um,
pure,
is very efficient, or yeah?
I'm a pure biologist, so uh, I took a picture of uh, pattern, geometric pattern. I input the image into Google search, Google image search, and in a second I hit uh, many similar pattern that suggest it's called a nematic liquid crystal. And I didn't know, I had never heard the term nematic, so I made a phone call to one of my friends in chemistry field, and I asked him what's nematic, what then what it means, and he gave me some advices about the liquid crystal. And so I'm not sure, but some people pointed out in the case of ordinary, usual liquid crystal molecules, the shape of the unit molecule is constant in size. Uh, but in this case, as we observe, the cell shape changes its long diameter moment by moment. So I guess this form of liquid crystal-like pattern needs a new model. And as some colleague pointed out to me, and asked me, uh, uh, we should study and construct a new model about this pattern. Okay.
The,
Yeah,
the li-
yeah, thank you so much for that answer. Thank you.
the liquid crystal makes me think that it might have something to do with the you said it was, I forget the name of the type of material that it was in. Uh, I want to say calcite, but I don't think that's what you said. Um, do you think it has something to do with that? Like it's taking on the mineral from its surrounding, and that may be the thing that's pushing it to do something.
Do you mean association with calcite mineral?
Yeah.
Oh, are you microbiologist?
No, I'm just a regular fellow, curious mind. That's pretty much it.
There is a field of biomineralization
Oops, sorry about the
and
sink. Sorry, I'm washing dishes. Sorry.
biomineralization, and I didn't mention this in the article because space is limited, but the evidence that that by I wanna I got I didn't mention in the article is this species has a strong ability to crystallize minerals, and I didn't publish this because of insufficient data for publication, but I believe this species crystallize minerals on calcite wall and form calcium oxalate due to its specific shape of a crystal. So, and calcium oxalate crystal formation is a signature feature of limestone cave associated bacteria such as streptomyces, which is most studied limestone cave bacteria. So, yes, this species might have evolved in association with calcite mineral. I think it's interesting, but it's another will be another research. Yes.
Have you had a chance to put to expose it to other minerals, uh, it slips my mind.
Yeah.
It's in like chalcopyrite
Yeah.
has
Yeah.
the
Pyrite. I tried once. I tried to submerge this colony in dense calcium solution, and I try to mimic calcite wall condition. Yes, and the other mineral I have not yet tried. Yes, pyrite sounds interesting. Yes.
uh,
You,
I
you know,
forget what. Go ahead.
oh, I was just gonna, some conjecture and speculation here. If the nematic crystal-like phase is formed by the elongation of each of the cells, but one environmental consequence might be that the channels will form in between those layers, and in a calcium-rich
Yeah.
environment, it's curious if the individual cells are contributing calcium to the channels. Would there be a calcium gradient that would maximize in the center and induce the function of, because the ejections seem to come from the center? It'd be curious to get the calcium um, distribution across the colony and see whether there's a particular gradient, and perhaps it's calcium that's signaling.
Yeah. Well, thank you for wonderful comments. Yeah. Calcium might be involved in the behavior. Yes.
You
Um,
might try
if
cyanide
I
too.
oh,
Like
please
for the
go.
chalcopyrite, you'd have cyanide within it within the sulfide. Uh, I know that's a terrible for us, but it does have a lot of manipulation properties to it.
Oh,
Thank you, Jake. Thank you, Jake. I think Katarina was trying to say something.
So sorry.
Oh no, please, please go ahead
Okay,
and
sorry. So
answer.
I'm just
And
looking
yeah.
for the
yeah,
mics.
no.
Yeah,
I'm,
sorry.
Then I wanted
I'm
to
very happy
to check.
to take such a wonderful advice. Yeah. but unfortunately, I left my laboratory. Now I work for another project, so I don't study now. So I miss so much my research activity. So if someone audience wanna let me continue this study, please contact me and please offer me such a position for research. I am ready.
Oh, we wish we have a lot of projects here in Science Society that we want to find funding
Now
for,
I work for the
and
project, educational project. I start up a new college in Bangkok, Thailand, and I kind of manage starting up a Japanese style education for engineers. It's a very very important project, and I will work for this Thai government for 2 years. But now I can't study. I have many online meeting with officers, but yeah, I I will be back to research.
I understand. Yeah, it's like getting paid for a wonderful hobby, right? To do research. Um, yeah, I wanted to check with you your time.
Okay.
If you still have time or if you need to go, because I know it's morning for you. You probably have
Yeah.
much work to do. So
I I,
I wanted to.
I took off time in the morning today, so
Oh, great. Yeah, if
no
you still have a few minutes, we for sure have more questions. But if you need to go, please let us know.
10:30. So I have a little time 30 minutes, yeah, and I have to go to
Okay.
the office. Not long.
Okay.
Yeah.
Well, and thank you for taking off the time to speak with us. It's such an honor. Thank you.
I'm very
I see. Oh, I'm sorry. There's a lag. I keep interrupting you. I'm really sorry about that.
oh,
Um, welcome, Yukiru. Yukihiro. Sorry for saying your name wrong. And did you have a question or comment?
No. I have been just listening about this dialogue, communication. Yeah, and I'm not a specialist in this field, multicellular something. Yeah, so somehow I come up to the speaker.
Thank you. We
Mm.
appreciate having you. Thank you for
thank you so
coming.
much.
And yeah,
Thank
Kate.
you so much. Sorry, Katarina. Yeah, I just wanted to say thank you, Yukihiro, and thank you for your comments in the chat. It's so special to have all of the guests who are here in the
Yeah.
room, as wonderful as it is. Thank you very much.
Thanks for mentioning about it. I'm not a specialist about a topic for today. One of those who are so much influenced by Carl Sagan's Cosmos, which was also popular in Japan in mid 1980s and mid 1980s and early 90s.
It's a great example of how we never know what the different things are that we will connect over, because Kuhui, you mentioned Cosmos and Dr. Carl Sagan, and then Yukihiro, has association with that as well, and me too, and probably other people in the audience. So it's just wonderful to see all the ways that we connect through science. So thank you.
Yeah, Carl Sagan's book translated into Japanese language. That's been and also sheltered in the
I'm sorry, I think you're breaking up in between. Oh, sorry. Now you're back.
sorry. The Carl Sagan's Cosmos translated in Japanese language shelved in the all small local Japanese high school, junior high school, elementary schools, in the libraries. But by that time, I was also one of those children who was influenced.
Now we can watch by
Yeah, thank
YouTube.
you.
It's from Carl Sagan that we all learned how to say billions and billions. I think that's everyone's favorite quote from him.
That's right.
And Kate, welcome. You have, I think, the last question. So please go ahead.
Hi, thank you all. Really good discussion. I think my question goes more towards the foundational aspects of storytelling, in that we see new scientific research coming out, but that is not always translated in terms of not only the media, but in terms of the everyday person. So, in your opinion, Dr. Mizuno, how do we communicate this? How do we translate this into a storytelling narrative? And also within that vein, there's been new research coming out of Australia that tells us actually that pessimistic research or storytelling, specifically within the sustainability space, the climate change space, has led to more empathy and more action. Whereas right now we are focused on more positive storytelling. And so I'm curious, in your perspective, how do we navigate that space? How do we communicate this outside the scientific concept and also within a very diverse world that leads to lasting change?
Thank you for asking me. Your question is about how good science material can be transferred to the people, particularly kids, who can understand that in their own language, right? Is my understanding correct?
Yeah, I think my question goes more towards how do we navigate the
Navigate.
storytelling? Yeah, how do we navigate that from the scientific
Yeah,
community beyond the media towards the individual
individual
perspective?
perspective beyond media. Beyond media.
I would like to contribute if I may.
Yes, please go.
Yeah. So we have here our scientist, our researcher, and we all have our different jobs or affinities or passions in life that we explore. And so, Kuwei, you have bestowed upon us this gift of your research and your passion. I work in arts and science education, and it would be, wait, Dr. Shaw. Dr. Shaw's doing some dishes too, maybe. For me, I might be the one to take and interpret this for students in a culturally sensitive way. And so Kuwei has come here as storyteller within science. And also, I don't know if you were here earlier, and he was talking about his path to this research and interests that he had. So I would say, Kate, I'm not trying to answer your question that Kate has asked you, but I also want to just remind people who are here, what exactly are you asking of our guests to answer? He's come here specifically within this science realm which also extends wherever we allow it to. So that's part of what we're doing here in Science Society is providing, trying to provide access for everybody to come and listen. And so that's how Kuhue is doing that through this app right now, is to make this science accessible.
Wow, thank you so much.
Yeah, I think that was a beautiful comments and discussion in the end. Yeah, and thank you so much for coming here again. I that's that's exactly our mission, and Kohei was so friendly to come here and his time and take off time of work to share his research in a way that we could all kind of follow, even if you were not microbiologists like I'm not. But and also everyone here that helps with this club, like Victoria, Katie, Serena, Dr. Shah, this team of wonderful people always come here and support and help with this mission. So thank you to all of you to do exact this, and we wish you a wonderful day, Kohei, and wonderful holidays, and maybe you can come back one day, and hopefully we will find you a way to
And thank you so much.
come back
Thank you so
to
much.
basic research.
Thank you so much inviting me and also introduce this wonderful place. I think it's a wonderful place to communicate. And as you say, we need this kind of place to deliver a specific scientific topic to the people with other background. And yes, that's the I understand that's the science society here. Thank you so much for having me.
Yeah, I also want to thank you, and just in the sentiment that Kate mentioned about storytelling,
Mm.
one takeaway from this, and to put a spin on it, is just how did the first cells learn to work
Mm.
together, and what is the origin of collaborative behavior as an advantage in survival? I mean, it's a kind of an interesting
Thank you.
take, but thank you for your talk.
Thank you so much.
Well, wonderful. We Yeah, thank you and have a wonderful day. This was a really interesting discussion. Your work is really amazing. And if you like discussions like this, everyone follow the club. We will have more discussions like this. Also this week we'll have for sure Dr. Ginzenberg talking about Hipparcos Star Catalog Revealed. It will be a really interesting discussion about his research. And then Dr. Eisen will come talking about microbiota and how they promote social behavior through microglia. Serena, you have to be there for that one. It's on Friday at 6
Yay!
p.m. Then on Saturday, hopefully Dr. Kabuku will talk about research shows how complex prehistoric like how complex prehistoric cuisine was through archaeology findings. So Serena, maybe your your boyfriend will come because I thought it was really interesting. They found like really prehistoric remains of like food stuff and found that they had quite complex recipes
Oh,
apparently.
cool. I just elbowed to the right.
And then on Monday, Dr. Young will come from UC Davis and talk about learned lensless 3D camera. Yeah, how he created a 3D camera without a lens through interesting technology he developed. And that will be the last room for this year. And then we will have many more rooms in January again. We already have a pretty full schedule until end of January. And so, yeah, I hope I hear you all back soon. And thank you, everyone. And a special thank you to you, Dr. Koe Mizuno. And I hope we'll stay in touch. Thank you so much. Okay, I'll close the room in 3, 2, 1. Bye, everyone. Thank you.
Thank you.